decades of basic research into the mRNA technology that went to it has made it to the global headlines recently, currently, and might be also in the future a lot of talking about mRNA. As I worked in the past a lot in basic research in the RNA field, RNA degradation mechanism, RNA control, translational control, technologies around RNA. I'm very much excited now about the breakthrough for mRNA technology also on the personal level. In my presentation now, I will describe the recent rise of mRNA with its revolutionary potential for biopharma. I'm happy to speak now, and if you have any question, there's a chance to ask your questions at the end of this talk. So when has this all started? The whole story of mRNA. It was in 1961 that an information carrier called mRNA was discovered by two groups in the Harvard University and Institut Pasteur. It was the same issue of Nature when the experimental results were published. And I think it's yeah, like already the codon was discovered. The ribosomes were known. That one knew that the codon translate into protein later, but in between, the RNA was still missing piece, and this was discovered then. But since its discovery, it was still a long way to mRNA as a therapeutic or vaccine. It was already described in 1961 that this is an unstable intermediate. So both describe that. And the problems were still there. And it took a long time till now because technologies needed to be developed, hurdles needed to be overcome, that it will end up in a solution for biopharma, in a solution for vaccines used, for therapeutics used. So mRNA was unstable. There was an immunogenicity problem when it was supplied, and there was still so many missing pieces also when it comes to delivery. So the mRNA vaccines were the beginning. This was the start when the rise of the technology came. And it came when we needed a solution fast in this current pandemic situation. So companies and researchers were speeding up. They're working hand in hand together to work on quick solutions in headlines like light speed, warp speed. So it was really, really a run. And it were all over in the news at those days. And the people were really hoping that this technology will save us. So this was the start of the revolution, as mRNA could already reveal the full potential. After decades of research were invested in the overall RNA topic when it comes to technologies, understanding transcription, translation, regulation processes, etc. So what makes mRNA vaccines strong? Why are they a new era in vaccinology? It's a quick and simplified production process in comparison to protein-based therapeutics or vaccines. I mean, it takes a lot of time to do cell culture and to set this all up, and there are so many insecurities in between. It can be quickly adapted. Like for viruses, we have it now with the SARS-CoV-2 virus. It mutates, and then you need a new vaccine at one point, and this can be quickly adapted as the process allows it. And mRNA is broken down very quickly in the body. So there is also a reduced risk for long-term side effects when it's applied to patients. And in many cases, no drug amplifiers adjuvants are required. And this also reduces the risk for side effects. So what kind of technologies needed to be developed on the way to enable this rise? One of them is the use of modified bases. studies with mRNA family members like the tRNA have shown the potential of modifications in RNA molecules and what they can bring. That they can, when you use a modified nucleotide, like the N1-methyl pseudo-UTP that you see here in the slide, it's one of the most popular ones currently, also used in the mRNA vaccines. They have the potential to reduce the binding of innate immune sensors. So these innate immune sensors, they are receptors in the body that recognize RNA when it comes. So it's a protection from pathogenic RNA coming from viruses. So the immune signaling, the innate immune signaling will take place. And these modified nucleotides can modulate this response, can reduce toxicity. And it was even shown that for some of these modified nucleotides, that it can also increase expression levels, so the translation into the proteins in general. What else needed to become like a hurdle to be overcome? It comes to the delivery system. So RNA is quite unstable. There are nucleases around that degrade it, but it also has to come through the lipid bilayer. And one technology that is quite supportive here are lipid nanoparticles. So an RNA negatively charged needs to overcome the negatively charged lipid bilayer, and lipid nanoparticles help here. Then it goes into the endosome, and then it also needs to go out of the endosome. So a good delivery system also allows endosomal release within the cells. And a good delivery system also allows the protection of the mRNA on its well, that it's not degraded, that pH is not a problem to the RNA. And there is still potential for future modification. So you can think about targeting options, about further modulation of immunogenicity as well. What other knowledge went into it to make it successful? It's about the mRNA construct design. A lot of basic research went into it to understand all the elements that make up an mRNA. So we have at the 5' end the so-called 5' cap structure. The 5' cap structure is important for the mRNA maturation, for the stability and the translation. So for example, for the translation initiation factors are interacting with it. Another important element is the coding sequence. So here it contains the genes to be expressed that you actually want as a therapeutic or active vaccine component. Codon optimization and modifications here of nucleotides can contribute to the stability and the translation efficiency. Then at the 3' end, you find the poly-A tail. The poly-A tail is at the 3' end, and the length is also being studied a lot, how it contributes to stability, but also translation efficiency. So a distinct length is required and needs to be taken care of in the overall design. And then you find here at the 5' and 3' flanking the coding sequence, you find the so-called untranslated regions. And these are often full of elements where RNA binding proteins are interacting with and influencing the translation efficiency or the localization of the RNA inside the cell and the stability. So currently, some of the known constructs coming from other mRNAs are used to design these UTRs to contribute to a stable RNA and to an RNA that is efficiently translated into the protein of interest. What are alternative mRNA construct designs that you can find? So I just showed a general mRNA, but currently a lot goes also into what is else in the mRNA technology possible. So it's possible to have so-called circular RNA. As I said, the stability is quite important to have the poly-A tail at the 3' and the cap at the 5', because open ends are a problem for an RNA to be not degraded. So there are mechanisms to degrade it. But if you don't have the 5' and 3' ends, then you don't have the problem of having this degradation thread within the cell. And yeah, this can be done by using like a splicing mechanism to use a ribozyme reaction to do the circularization through the RNA itself, or there are also mechanisms to use it, use enzymes to do a ligation of RNA to circularize it. And then, as I said, the cap structure is usually important for translation, but you can also work with an internal ribosomal entry site at the 5' end. This is coming from some of the endogenous RNAs, but also known from some viral RNAs carrying these internal ribosomal entry sites. These are usually quite structured elements that you find at the 5'. It's cap-independent translation possible. You get rid of the cap structure. Other RNAs are self-amplifying RNAs. So it uses a replicase system coming from alpha viruses. You can use it in cis or in trans. So cis means on the same construct, trans on two separate constructs, and it allows that the RNA itself amplifies it as soon as it's in the cell. So there is an amplification in the cytoplasm, so you can use less RNA. So currently companies are working on this as well. So what are all the advantages coming with the mRNA technology to make it revolutionary? Therapeutic protein production is a laborious process. There is so much uncertainty here because you have to set up it for all the proteins. You have to have purification process. You need cell culture, etc. mRNA is produced in a standardized one-way reaction using the same protocol, regardless of the coding sequence, making it a more robust, and yeah, you can also anticipate the risks a little bit more. Misfolded or improperly modified proteins can cause immunogenicity and adverse effects. When you have your synthetic mRNA, you design it in a way that it transiently delivers the proteins of interest directly in the cell. So the body is producing your therapeutic itself. The process complexity of protein production complicates GMP compliance in biopharma. With the mRNA technology, this allows a simplified GMP production and reduced development costs. Then what can it be used for? So mRNA is a platform technology. It can be used for various applications, coming from vaccines. You can also go to protein replacement, to gene editing, CRISPR-Cas, personalized medicine, cellular therapies. A lot of that is currently worked on. So for example, for CRISPR-Cas, you can express the Cas9 maybe as an mRNA and deliver it. Or for cellular therapies, like the CAR T cell therapy, you can maybe use an mRNA that is expressing a T cell receptor targeting activated fibroblasts, just as some of these examples. So now here you can see the overall and general synthesis platform and which steps are usually followed. So it's just a general overview for you. At the beginning, you have a template DNA. This can be a plasmid DNA, it can be a PCR product, it can be a cDNA. And usually it carries a promoter sequence for an RNA polymerase. When you have a circular construct like the plasmid, you need to linterize it to have an efficient in vitro transcription reaction later on. And you can use a restriction enzyme like XBAR1 to linterize it, and then the in vitro transcription reaction is following. There you need an RNA polymerase. This can be a T7 or SP6. Yeah, it depends on the promoter that is in the DNA used. And then you also need ribonucleotides or pyrophosphatase to make it more efficient to remove the pyrophosphates coming in the reaction in between and inhibiting the T7. So you use pyrophosphatase, you use different buffer components. This can be spermidine, magnesium chloride is quite crucial. All this is added, or HIPAA buffer, tris buffer, or DTT. And in this reaction, you can decide if you already do here the modifications. So this can be done during the reaction, or it can be done afterwards, so post-transcriptional modification. With modifications, I also mean the poly-A tail and the cap structure. This can also be added by enzymatic mechanisms, by using, for example, a poly-A polymerase or capping enzymes for the cap structure. Then quite crucial afterwards, the purification steps. So the plasmid is digested by an enzyme called DNase1, and the proteins are removed using proteinase K. And then the RNA is purified using chromatographic methods. It's also used to all the impurities that can also come as byproducts as well. And then for the final formulation, as already mentioned, you need a good delivery system, but also something to stabilize it. And this can be done like with lipid nanoparticles as well. So what are potential contaminants and byproducts in the mRNA manufacturing processes? This can be double-stranded RNA. This is a byproduct during the RNA polymerase reaction. Double-stranded RNA, when it's recognized by receptors like RIG1 or toll-like receptor, this induces immunogenicity. So the innate immunity is pushed on. And this is not a good reaction to have for some applications. And if you want to modulate it, you have to choose the right reaction condition during the in vitro transcription reaction, but you also have to be sure that you're using purification methods that are also removing these impurities to some extent, or to the best extent for your purposes. Metal ions are a threat to RNA, as they can also cause cleavage of RNA when it's also at elevated pH. And metal ions are also a potential risk for patient safety. So for biopharma purpose, you don't want to have them. So please reduce uncertainty. You have to choose the right raw materials that are also tested for those impurities to reduce any uncertainty here. And then there are the RNases. They're all over. Everyone already worked with RNA knows they're in dust, they're in air, it's everywhere. This can cause rapid degradation of RNA. So you can, go around that by using RNase inhibitor during your preparation steps. You can use tested raw materials that are testing for nucleases, and you can also work in a clean environment to reduce all the risk that your RNA is degraded on the way, and you are losing material and you're getting bad material as well. So bridging now the gap between laboratory and industrial scale. Manufacturing process must be adapted to GMP conditions if you are active in biopharma. And there you have to adapt to narrow product consistency, quality, and safety tolerances. You need certificates, you need lot-to-lot consistency, etc. And then the right synthesis setup needs to be taken, and the purification processes have to be chosen. You have to control the impact on immunogenicity and stability profiles. And you have to choose the right raw materials from the start to the finish. It also accelerates when you're not switching in between the time to market by using the right quality, like animal origin free. It's required for biopharma currently by regulatories, antibiotic free, to keep the risk low for patients, and also to have extended impurity testing for heavy metals, bioburden, etc., and to have it at GMP grade from R&D to commercial stage. Avoid reagent changes and minimize the regulatory challenges. What do we do from Roche Custom Biotech to support you on your process when you want to create something for biopharma, like a therapeutic mRNA based? We do this extended impurity testing to limit introduction of host cell impurities, heavy metals, endotoxins, into the drug manufacturing process. We work according to GMP grade and ISO standards to rely on validated methods and processes, as well as appropriate documentation, facilitating good regulatory compliance. And most of our materials are also antibiotic-free, or at least beta-lactam antibiotic-free, to minimize the risk of potential allergic reactions. We also have animal origin-free material to minimize the risk of viral contamination in the therapeutic at the end, risking patient safety. And it needs to be scalable, so we can produce in higher amounts, and it's also consistent lot to lot to allow consistent and commercialization of the drug that is produced. So this is the regulatory disclaimer in case we are relating to our products. And I want to thank you for your attention, and the mRNA revolution is just beginning. So please join us. And I was very happy to give today this presentation. And I'm also very happy if you have any questions, even after this talk, like not questioning now, to contact me directly, give me a call. You can also experience what's there on our website. So a lot of this content I was just presenting, you also find on our website. And I already mentioned my colleague Sebastian. He is currently in the auditorium and happy also to assist you in case you have any question. So here are also his contacts, and you can give him a call, write a WhatsApp or an email. He is happy to discuss the topics with you as well. And for everyone else, we also have, we are working from globally, having teams in different sites and in different regions. So you can also reach out to your specific regions when it's not Latin America. So thank you for your attention, and please let me know if you have any question.